Action potential can be defined as a sequence of changes in the membrane potential in an excitable cell due to opening and closure of different ions after the application of threshold stimulus. And action potential in cardiac muscle is of two types: fast response action potential and slow response action potential. In fast response action potential is seen in working myocardium, that is atria and ventricles and Purkinje fibers, and it is characterized by rapid depolarization. When the firing level is reached, there is a very rapid change in the membrane potential, producing a sharp upstroke which is called as zero phase. The resting membrane potential of ventricular musculature is about minus 90 millivolts. In this phase, the membrane potential crosses the zero line, that is positive overshoot. The inside of the cell becomes positive with respect to outside, and the cell is fully depolarized. The initial depolarization is due to sodium influx through rapidly opening sodium channels. In initial rapid depolarization after the zero phase, uh, there is a slight fall of membrane potential towards zero potential line, and this is named as phase 1. This phase is due to closure of fast sodium channels and opening of potassium channels. That means efflux begins here. Then we have a plateau phase. After phase 1, the membrane potential is maintained for some time at that level, that is, the repolarization is prolonged. This phase is named as phase 2. It is due to the opening of slow calcium channels along with potassium efflux. Thus, the potential change due to the potassium efflux is neutralized. Late rapid repolarization. When there is a rapid repolarization at phase 3, this is due to the closure of slow calcium channels and potassium efflux. Baseline, that is the polarized state, is the last phase where the RMP is restored by the activation of sodium potassium pump, which is caused by potassium efflux. So these are the different phases of action potential in the cardiac muscle. There is a zero phase which occurs because of the rapid sodium influx. The phase 1 occurs because of the potassium efflux, and the plateau phase occurs because of the continued potassium efflux and long acting calcium channels, entry of long acting calcium channels. And again, phase 3 occurs because of the continued potassium efflux. Another action potential is slow response action potential. This type of action potential is seen in the pacemaker tissues. Before the sharp upstroke, which is seen in myocardial cells, there is a gradual slope here. In sinoatrial node and AV node, the depolarization in the beginning is gradual and RMP is automatically approaches to minus 60 millivolts. This phase of gradual slope is called as pacemaker potential. When this pacemaker potential reaches the value of minus 60 millivolts, firing occurs. Now there develops a sharp upstroke, and there is no plateau in action potential of the SA node and AV node. The ionic basis of slow response action potential is at the peak of each impulse, that is end of the depolarization, potassium efflux begins efflux brings about repolarization. Then there is a gradual decrease in potassium efflux followed by the opening of transient calcium channels, and there is calcium influx, and this accounts for prepotential. When membrane potential reaches the firing level, there is opening of long lasting calcium channels and there is a calcium influx. When the membrane potential reaches the plus 35 millivolts, there is a closing of calcium channels and opening of potassium channels. Note that electrical and mechanical response overlap in cardiac muscle in contrast to skeletal muscle. This diagram showing the action potential of slow response type of action potential in AV node or SA node. So the initial phase is not rapid, but it is very slow, which is called as a pace pre-potential till it reaches the threshold level. The ionic basis is for the pre-potential. In the initial part, it is because of the delay in the closure of the uh, potassium channels, that is closure of potassium channels. Then the the transient calcium channel, which causes the entry of calcium, and then there will be development of the threshold, so which leads to long acting opening of the long acting calcium channels, that leads to the depolarization, and the repolarization is caused by potassium efflux